All right, so today we're going to go over what a high school lab report, science lab report should look like, okay, what the expectations for you are. I'll go over the grading kind of expectations or rubrics so that you understand what I'll be looking for when I grade it.
Are we doing the lab tomorrow?
We are going to do some of the pre-lab stuff tomorrow and finish it up Friday, and then we're actually performing the lab on Monday. All right, we're also going to talk a bit about like lab behaviors, expectations and stuff for safety within the lab itself. Okay, so first things for labs, safety-wise, okay, if you have long hair, please make sure that you put a hair tie in your science binder, okay, so that you can tie your hair back anytime we go into the lab, because it does need to be out of your way, so you don't like lean forward and get it in something. Okay, especially if it's an acid or something like that, then the whole lab will smell like burnt hair and your hair will look terrible, and that would all be bad. So make sure you have something that can tie and hold your hair back. Okay, similar with clothing. Okay, if you have anything that's like very loose that you're wearing, you might want to leave that behind just so that it doesn't go into something when you lean forward. If you're wearing a hoodie or anything that has drawstrings of any kind, make sure that those are tied up and tucked in, okay, so that they don't, again, fall into anything. You will, of course, be required to wear goggles. Okay, now we have some of the classic big giant things that fit over top of prescription glasses, so if you do wear prescription glasses, you will have to wear the larger ones, because prescription glasses on their own do not cover from the side. Okay. If you have prescription glasses and contacts are a good thing to wear on lab day. Okay, that way you can wear the smaller goggles, and they're not quite as obtrusive, and they don't leave big marks in your face, okay, which do tend to last for a while. So you will be required to wear those. As soon as you go into the lab, you put them on, and they don't come off until I tell you to take them off.
Will this stuff be over quiz tomorrow?
No. Okay. Also, footwear. Okay. You do need to have shoes on that entirely cover your feet. Okay. Sandals, flip-flops, open-toed shoes, stilettos. Okay. Things like that are not appropriate shoes for the lab. Okay. You need flat-soled shoes that cover your entire foot, preferably with socks on inside. Okay. If anything were to spill on the lab table and then run off, it could get on your feet, and if they're not properly protected, okay, then we have an injury and an accident. Okay, so make sure you have proper footwear for the lab as well. Okay, that's really it in terms of like clothing and stuff. Safety behaviors within the lab are also very important. Okay, the lab is just not a place where we can horse around or joke around with each other. Okay, we could be dealing with dangerous chemicals, and we are. Okay, so we do need to be mindful and respectful of that. Okay, so no pushing, shoving, playing a joke on somebody and then they get startled and they spill stuff. Okay, all that stuff that gets left in here and it doesn't happen in the lab. Okay, there's a zero tolerance for that. If I have to talk to you about it, you'll be in here, not doing the lab. Okay, and depending on the seriousness of the infraction, it may be your last lab. Right, because safety is paramount. Okay. No food or drink, obviously, in the lab. Okay, that's another one. Sometimes people are like, they've got an electronic version of their lab report on their laptop or device or something like that. If it's your phone, that's probably fine because you can take it in and out of your pocket, but I wouldn't recommend bringing like a Chromebook or a laptop or something like that across the hall. I think it's spilled on that, it's toast. And there's a sink on every lab station, and water is always splashing out of those. So it's probably not a good idea to have anything electronic that you can't put in your pocket and keep safe. Okay? Hand washing. We're going to do that, okay? Especially after the lab. Okay? Make sure that you wash your hands thoroughly, okay? Soap and water upon finishing. And then there are paper towels at the back of the lab. So the lab is actually just across the hall here, okay? And it's just the sink and the main hand wash sink and paper towels are right there on your left as you go in. There are also lab aprons. Those are not mandatory, okay, but if you do want to wear a lab apron to protect what you're wearing, that's not a bad idea. Just know that they don't cover everything, okay, like they're fairly narrow at the top. They kind of go down right about out here and then they spread out under your chest to tie around in the back, right? So if I were you, I wouldn't wear it like, you know, your $1,000 Armani tuxedo, okay, or your, you know, cocktail dress or something like that to the lab, okay? That's probably not the best choice for what to wear. I would, you know, have something, you know, that's maybe not your favorite for lab day, just in case. Okay, if anything happens, and sometimes it does, and sometimes it's just purely by accident. Someone drops a thermometer and breaks on the floor. That's fine, as long as you don't break two in one day. Okay? If something breaks, something happens, something spills, we'll take care of it. Everybody just stays calm. Okay? If you get something on you, okay, then you have to go immediately to the sink and keep your hand, if it's on your hand, under the water, cold water for 5 minutes. Okay, just to be on the safe side. Okay, it's usually on people's hands, but if you get it somewhere else and you have to go to the bathroom to take care of it, go right ahead. Okay, if anyone for some reason gets anything in their eyes, okay, that's where I need everybody to clear out. Okay, because I will have to help that person, because when stuff gets in people's eyes, they cease to be reasonable and cooperative. Okay, they just tend to scream and flail. And so I'll need everyone to clear out while I grab that person and take them to the eye wash station. Okay, and I just sincerely hope it goes better than the last time I had to do that, where we both ended up bloodied. Okay, so that was very bad. Because the person was like flailing around and I was trying to get in to help them and they smacked me backhanded right across the nose. So I'm leaking and then I'm upset. Grab a handful of hair and jam the face into the eye wash station. Well, there's a little, like, the eye wash station has those two little things that look like eyeballs that shoot the water like water fountains. And there's a bar right across the bottom, and I got them right in the nose. They're both leaking. So yeah, it didn't end well. But if you wear your goggles all the time until I tell you to take them off, none of that will have to happen. Okay, so that's generally the best bet. Okay, I'll remind you of that stuff when we get in there, and if there's any other cautions that are appropriate for a particular lab, obviously I will tell you those as well. Okay, so looking at the format for the lab report, we are going to follow the scientific method to the letter. Okay, so every lab starts with the problem that we are investigating. All right, on a typical lab report, okay, I will give you a template. Okay, the templates typically look like. I'll show you the one we're going to be doing here. Oh yeah, right. Not the right one. Okay. Sorry guys, get that open here. Okay, so when I give you a lab report to do or get in Google Classroom, you will get a template. Okay, so it's kind of pre-built, but there's not much in it. Right? You're going to have to fill in everything. Right? So there should be no worry of missing any parts because all the parts are there. Okay, you just have to fill stuff in. So you'll get a Google Doc. Okay, and instead of saying U1 Properties of Compounds Lab Report, it'll say your name. U1 Properties of Compounds Lab Report, okay? And that will be your lab report that you will hand in to me electronically on Google Classroom. Okay, everybody follow there? So it'll be set up like that, and that's what you'll be working from. Okay, so every lab report starts with a problem, all right? The problem is not going to be in there, okay? I will tell you what the problem is that we're investigating, and then you will have to put it in your own words in the form of a question, like Jeopardy, okay? So if we're looking at kind of a simple experiment where, let's say we wanted to see what the effect of light is on plant growth, because obviously that's very simplistic, but let's just use that as our example. The problem could be, what is the effect of light on plant growth? It doesn't have to be anything earth-shattering or like 4 pages long. It just needs to be a simple sentence, okay, in the form of a question that says what you're investigating. Okay, everybody follow there? All right, the next part is the design. Okay, in the design, you are going to identify your manipulated variable, responding variable, or variables in some cases, okay, and 3 control variables. Okay, so you're going to identify them and you're going to explain them. All right, so just a quick review. The manipulated variable is the part of the experiment that you as the experimenter are changing. Okay, it's the thing that you are manipulating. The responding variables are the things that you are observing or making measurements of or whatever. In some way they're going to change as a result of what you did. Okay, and they're the things you're going to be watching. Your controlled variables are any other things in the experiment that could cause a change in your responding variables if you didn't keep them the same all the way through or for all the subjects. Okay, everyone kind of follow me there? So if we're looking at our experiment here about the effect of light on plant growth, my manipulated variable would be the amount of light. Okay, that's what I'm trying to study. Right? Now, if I'm going to do a good job of explaining that in my lab report, I'm not just going to go the light because I change it. Okay, that's not a high school lab report level of detail. Okay, so we would be looking at something like this statement here. The amount of light given to plants is the manipulated variable because all subjects will receive different amounts of light and light powers photosynthesis. So in that statement I've said, here's the manipulated variable, here's how and why it's being manipulated. Okay? This is what I'm studying about it. This is how I'm manipulating it. Okay? It doesn't have to be super detailed. Like I didn't say one plant's going to get this many hours of sun, this plant's going to get this many hours of sun. That's for the procedure. I can put that detail in the procedure part. Okay? As long as I've indicated here that there are going to be several plants, all that get different amounts, that's good enough. Okay? Everybody follow there? The responding variable, okay, in this lab, very simply, is going to be the thing I'm watching for. Okay? Which is going to be the plant growth. Okay? Now, this probably isn't as thorough as it would need to be. Okay? The growth of plants, because plants getting more light have more energy for growth. I would also indicate my measure of growth here. Are there a lot of different ways to measure how much a plant grows? Yeah. Okay? I mean, I could measure the height, but that's only one way a plant grows. Okay? Yeah, I could measure the mass of the plant. Okay? You know, how much more tissue does it put on? If it's a plant that produces fruit, okay? I could also look at the amount of fruit it produces. If it's something like grain, I could look at the yield of the plants at the end, something like that. You would probably want to have more than one measure for the growth of a plant, and you would indicate that. Okay? So you'd say the growth of the plants as measured by, and then list these things, okay? Height, mass, those would be probably your best ones, okay? And then you might even have some qualitative observations, as well as the appearance of the plant over time. So maybe you take pictures of it every day, okay, and do a time lapse of it when you were done. Okay? And then I've also got in here how I think it's going to respond. So I've said the plants that get more light are going to have more energy for growth. Okay? So there's again a how it's responding and a why it's responding. Now, your controlled variables, that's usually the hardest part because that requires us to really understand what's going on in the experiment. I have to know a fair amount about plant growth to know what other things might affect the growth of a plant. Because I have to make sure those stay the same for all the plants. Okay? If I just say, okay, this plant's going to get no light, this plant's going to get 4 hours, 8 hours, 12 hours, 16, 20, and 24, and I have those plants, okay, and then I just randomly give them all different amounts of water, am I running a controlled experiment? No. Some of those plants are going to grow better because they got more water. Okay? I don't know whether the plant that got 0 hours of sun also didn't get any water. Well, then what killed it? The lack of water or the lack of light? I don't know. Okay? So controlled variables are the other things that need to be the same so that they affect the plants all equally, or the subjects of whatever test you're running. So as an additional example, when pharmaceutical companies are testing new drugs, okay, so let's say they're testing like a new allergy medication or something like that, okay, they will have several test groups, probably all to get different dosages of the drug, and then they'll have a control group. That control group gets a placebo. They get a sugar pill, okay? Because there's something in pharmaceuticals called the placebo effect. Sometimes just giving somebody a pill makes them feel better. Okay, it's that whole mind over matter thing. Okay, so you have a control group of people who get a sugar pill so that they don't know they didn't actually get the drug. That way everybody gives you honest responses when you're asking them how do you feel. Okay, if you've got an experiment and at the end of it all your control group people said, yeah, my allergy symptoms were way better, your drug doesn't work. Okay? All you got was placebo effect. Okay? Everybody felt better because they took a pill. Okay? Now, if your control group is sneezing and they're crying and they got watery eyes and they feel terrible all the time and all the people who took your drug feel better, then you're onto something. Okay? That's the purpose of a control group and having variables like everybody got a pill, okay, be controlled. Okay? It can affect the experiment. Does that sort of make sense? Okay? So for an experiment on plant growth, we would want to make sure all the plants get the same amount of water. All the plants get the same type and amount of soil. Okay? That they're all getting the same kind of light. I don't want some plants outside getting natural light and some plants under a growth lamp. That's not the same kind of light. All of those things would be things I would have to control. Okay? So, your controlled variables are the other factors that could affect the outcome of the experiment if left unchecked. So, first controlled variable, same species of plant. Yeah, that's kind of important. Okay? I can't compare a lodgepole pine, a carrot, and a bean. Okay? They all grow at very different rates. Okay? So that would hardly be a fair experiment. So, different plants grow at different rates. Same amount of water, because water is an important factor in plant growth, so each plant needs the same amount, so it can't affect the growth rate of the plants. Same soil. Plants get their nutrients from the soil, and different soils hold water differently. Keeping them the same ensures it cannot affect the experiment. Same temperature. Okay? Plants typically slow their growth in extreme heat or cold. Choosing an effective temperature for all plants ensures that it does not affect the experiment. Okay? Basically, you would have to do this under artificial light. There'd be almost no way you could have a controlled experiment with sunlight, because sunlight is going to affect the temperature. Okay, so you would probably have to have like LED lights that produce almost no heat in every plant in a temperature controlled box. Okay, or in this room, and then you just put something over them so that they don't get any light, but they still get the same temperature. Okay, so you don't have to list that detail in the design part, you'll list it in the procedure. Okay, but you're telling people, here's a controlled variable, how I'm controlling it, and why I'm controlling it. Okay, so have you noticed that every single variable has a how and a why? Okay, really important that you do that. Okay, what I often see on people's lab reports is manipulated variable, light, responding variable, growth, controlled variables, water, soil, and temperature, and then there's no explanation. That's a 2 out of 5 at best. Okay? The explanations are important. All right, that takes care of the design part. So the design part is out of 5 marks. Okay? That includes your problem, okay? And it includes the variables all being identified and explained. The explained part is the most important part. Okay? Everybody with me there? Okay? So the first part of that lab is out of 5. The design part is out of 5. Okay, what comes after that? Your hypothesis. Okay, up until now, you've probably been taught that a hypothesis is an educated guess, which, you know, in junior high is probably okay. But we don't want to guess in science. Okay, a hypothesis should not be a guess. Okay? A hypothesis is what we call a hypothetical deductive statement. Okay, it's not a guess and it's not a question either. It's a statement. Okay, it says that if this relationship is true, and an experiment like this is run, then these will be the results. Okay? It has to predict the results so that you can unequivocally say at the end of the experiment that your hypothesis is acceptable or that it has to be rejected. Okay? So you can't sit on the fence in a hypothesis. You have to put yourself out there and say, this is what's going to happen if I'm right. And then if you're not right, then you have to say that in the conclusion. Okay? And it's totally okay if your hypothesis ends up being rejected. In real science, most are. Okay? And then a new experiment is designed, and so on and so on. So you would never lose marks in a hypothesis if the hypothesis ended up being wrong. That's perfectly fine. Okay? So if you end up having to reject the hypothesis, you won't lose marks for that. Okay? As long as it's in the right format, that's okay. If in the conclusion you say the data does not support this, therefore it's rejected, that's all right. Okay, everybody with me there? Okay, so since a hypothesis is a hypothetical deductive statement, it has to contain 3 words. If, and, then. Okay? Every hypothesis should contain if, and, then. Okay? If this is true, and this experiment is run, then the results will be this. Okay? If, and then statement. All right, so for this lab here with the plant growth, okay, my hypothesis would probably sound something like, if plants require light in order to carry out photosynthesis and grow, and an experiment where plants are given different amounts of light over a certain time period, then the plants that get the most light will show the most growth. Okay, have I put myself out there? I have. I've said more light means more growth. Okay, I didn't sit on the fence and go, oh, well, you know, some plants will do good and some plants won't. Okay, I can't really say that. I'll definitely be right. Some plants are going to do good and some plants aren't. But I have to say which plants are going to do well, okay, and why. Okay, so your hypothesis, 5 marks on your lab is your hypothesis. Okay, 5 marks. If, and then statement that communicates what the relationship between the manipulated and responding variables is. Okay, that is the if part. Okay, briefly describes the experiment. That's the and part. Outlines the expected results, that's the then part. Okay? All right, so our example here could be if plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. Obviously, this is a pretty simple experiment, so it's also a pretty simple hypothesis. Okay? All right, so just a reminder, if is what the cause of the problem or the relationship being investigated, and is a brief description of the experiment. And remember, it's brief, okay? You don't have to go into every plant is going to be in a 4-liter pot and they're going to get this many grams of soil and blah, blah. That's for the procedure, okay? It doesn't need to go in that part, just brief. And then the then part, what to expect if you are correct, okay? We generally try to avoid the use of I, us, or we in a hypothesis, okay? Notice that in the example hypothesis, okay, none of those words appear. If plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. That makes the hypothesis repeatable. Okay? That means someone else can try to verify your results by performing the experiment themselves. They don't require you. Okay? Because if you go, then we will be able to do this, or I think this, now you have to be present every time that experiment's repeated, because you were in the hypothesis. Okay? So I see this all the time. Okay? A hypothesis should never say, I think. Okay? This is going to sound really mean. I don't care what you think. Okay? I care what you can prove. Okay? That's what science is about. Okay? Here's the relationship. Here's what's going to be done to test it. Here are the predicted results of that experiment. Okay, so we don't have any I, or we, or us. Okay, questions so far? All right, so like I said, 5 marks for the design, 5 marks for the hypothesis. Okay, so the hypothesis is a pretty important part of the experiment. Okay, all right, then you're going to have your procedure, which includes your materials. Now, on most of the lab reports that you do, okay, that's already going to be in there. Okay, in this particular lab, we're actually going to design the procedure, okay, the one we're going to be doing on Monday, but the materials are all already listed here, so you're not going to have to put any in there, unless I say, oh guys, I forgot we're adding this thing, and then you have to type it in. Okay, these are all of our materials. Most of them are the possible identities of the unknown materials, and then a few pieces of equipment like pH strips, a conductivity apparatus, a scupula, okay, a depression plate, and thermometer. Okay. So in your procedure, okay, that's going to be the instructions for how to perform the experiment. Okay, so detailed, step-by-step, explicit directions for preparing and performing the experiment. Okay, so the preparing part would be the list of materials. The performing part is step 1, do this. Step 2, do this. Step 3, do this. Now, how detailed should a procedure be? Very. you have to assume the person who's following your instructions is a complete and total idiot. Okay? And that you do not want them making any decisions for themselves. Okay? If you are not explicit in what to do in every step, they will mess it up. That's what you have to assume. Don't take that personally when you do my labs, and I have the procedure already written out for you. Okay? But that's what you have to assume when you're writing the procedure, because if you leave any room for someone to come up with their own ideas, then the experiment isn't repeatable anymore. Two people could do it differently, and then they could get different results. That's not what we want. Okay? The directions have to be very explicit. Leave no doubt. That's what explicit means. It doesn't mean full swear words. Okay? It means that it leaves no doubt. It is abundantly clear what you want done. Okay? So if we're talking about this procedure here for the plants, probably your first step would be acquire the materials in the materials list. Okay? And then your next step would be fill each of the pots with this many grams or this many liters of soil. Okay? Plant the seed to this depth in the soil. Water the soil with this much water. Okay? Place the plants in these conditions. Okay, so plant number 1 will go in a room that never had, or go in a box that never has any light on. Plant number 2 will go in a box where the light will be on 4 hours a day, a timer is set. Okay, plant 3 will go in this box, and so on and so on. Be explicit. Okay, tell them exactly what you want. All right, and then tell them every week water the plant with 250 milliliters of water. Again, be explicit. This is how much water. Should I also say what kind of water?
Yep.
Yes, okay. I should probably say like tap water. Otherwise someone might use distilled water or feed them with bubbly or whatever. Okay, you want to make sure they're using specific stuff. Okay, even with the soil, you might want to specify buy like 5 bags of this type of potting soil and make sure that all of the pots get exactly the same amount of that kind of potting soil. If you just say put dirt in the pots, someone could go out and dig 5 different holes. Okay, well then they got 5 different soil compositions. That's no good either. Okay, be explicit, right? And the other thing you have to tell them in a procedure is what things to observe. Okay, so in this experiment you would probably say, on a weekly basis, measure the height of the plants and place them on a scale recording their mass. Okay, and you're telling them also how often to do that. Okay? And any other observations that you think would be important for the lab, you got to tell that person. Otherwise, all they're going to do is do the lab, and then at the end they're going to go, okay, I did it, and they're not going to have any data because you didn't tell them what to record. Okay? I know that seems like you're dealing with like a 6-year-old, but imagine you're dealing with a 6-year-old. Okay? That's the kind of procedure you want to design. Okay, so from the procedure, okay, and in this lab, your procedure will be 5 marks, but after this lab, you won't get any marks for procedure because I'll have written it for you, okay, so it won't be part of it. Okay, then you're going to have your observations. Observations are generally out of 3 marks, okay, and those 3 marks are based on, are the observations correct, okay, are the observations thorough, detailed, okay, so what kind of observations are you making? So in a lab on plant growth, okay, I would want to see a weekly height report and a weekly mass report for each of the plants. Okay, that could be in a chart. Okay, and maybe you have your pictures of the plant as well every week as part of the report. Also good. Okay, from your observations, okay, your observations lead to your analysis. Okay, analysis is 5 marks. Okay, and so those 5 marks are going to be about, basically the questions that are being answered. Most of the time your analysis is going to be, here's some questions, answer them. That could be performing calculations. In this lab, it's going to be getting the identities right. Okay, so A through K, did you get, was A water, was B table salt, was, whatever. Getting those right, that's your analysis for this lab, okay, out of 5 marks. Right? Should your answers be detailed if explanation is required? They should. On this lab, there's not really any explanation required. It's just a chart you're filling in that says A was this, B was this, A was ionic, C was molecular, whatever. Okay, it's just a chart you're filling in. Okay, and then from your analysis, you have your conclusion. In the conclusion, it is expected that you will copy and paste your hypothesis. Okay, so you just go right back up to the top, Copy your hypothesis, paste it down in the conclusion. Okay? And then you have to tell me, does your data support or reject that hypothesis? Okay? And that's gonna mean, for this plant lab here, let's say that our observations for the plant growth lab were this. Plant that got no light died. Plant that got 4 hours of light only grew a tiny little bit. Okay? The plant that got 6 hours grew reasonably well. Plant that got 8 hours grew reasonably well. Plant that grew 12 hours grew really well. Plant that grew for 16 hours grew exceptionally well. 20 and 24 died. Okay, so if my observations are that the plants that got the most light died, is my hypothesis acceptable? It is not. Okay, so I would have to write in my conclusion something like this. The observations were that plants receiving either very low amounts of light or very high amounts of light, 20 to 24 hours, died. Okay? Plants that received between 8 and 16 hours did very well, with the plants at the 16-hour end being the plants exhibiting the most growth. Okay? This means that a hypothesis stating that more light means more growth is not entirely correct. Okay? And I can say that, not entirely correct. I was right to a point. I don't want to say I was right, okay? But I can say that the pattern appears to be that light is an important factor in plant growth, but there's kind of a drop-off. There's a maximum amount of light that is healthy for a plant, and beyond that point, the plant is incapable of surviving. This number seems to be around 16 hours. Okay? That can be my conclusion. It says I have to reject my hypothesis. That's okay. But it also states my conclusion is that, well, my hypothesis was wrong. The data does say that there is a general trend of more light is good to a point. Okay? Does everyone kind of follow me there? The worst thing you can write for a conclusion is, I was right. The second worst thing, I was wrong. Okay? You weren't right or wrong. Your hypothesis is either acceptable or it's not. And you have to explain that. Okay? Your conclusion is 4 marks. Okay? You have to copy and paste your hypothesis, that's 1 mark. Okay? And then the other 3 marks are for, did you correctly arrive at a conclusion? Okay? Did you say the hypothesis is not acceptable and here's why, or the hypothesis is acceptable and here's why. Okay, that part needs to be in there. So your conclusion is going to be, a few sentences for sure. All right, and then the last thing, evaluation. Okay, in your evaluation, you need to identify 2 sources of error. Okay, and you have to explain to me how they affected the experiment and how you could correct them in the future. All right, so for this experiment here, one of the main flaws in this experiment was that we didn't really monitor the temperature of the plants. Okay? Light sources always produce small amounts of heat. Okay? That means that the plants receiving more light may have also been warmer. And the warmer the temperature, generally the greater the rate of photosynthesis. Okay? Therefore, this could have produced problems in the data. It may have caused the plants with 20 and 24 hours of light to die because they overheated. Okay? So in the future, a climate control system may be required to get better data done. I said how? I said this was present, this was noticed, this is the effect it may have had, and this is how I would fix it in a future experiment. Okay? Everybody with me there? Okay, another source of error, you could have, anything that was kind of beyond your control, okay? So maybe the power went out, okay, 1 day during the experiment, okay? Is the power being out for 1 day in a month-long experiment the end of the world? It's not, but it happened, and it was beyond your control, so it needs to be listed, okay? So you can say there was a power outage on day 16. None of the plants received light that day for a period of however many hours, which then maybe only affected a couple of them because maybe some of them were already in the dark when the power went off. Okay, but it's still something that happened and it needs to be listed. How do I fix that in the future? I have an uninterruptible backup power supply. Seems excessive, but that's how you could fix it, okay? Your fixes don't necessarily need to be realistic, okay, or cheap, okay? They could involve requiring hundreds of thousands of dollars worth of equipment and facilities, that's fine. It's still a way to fix it, okay? Your sources of error can never be human error, okay? And what I mean by that is this, I get this all the time in lab reports. My partner and I messed up sample 5, and we didn't get good data for it. Oh, so you're telling me you screwed up, and you were too lazy to do it over again? Is that what I want to read at the very end of your lab report? Okay, was that a correctable error at the time? Then correct it at the time. Okay, human error is not a source of experimental error. We're looking for procedural, conditional errors that are beyond your control. Okay, something that's in your control should be corrected at the time. Okay, so never list human error. Like, oh, I forgot to do this part of the procedure. Well, then the whole thing is irrelevant. Okay, now I've got to go back and take marks off for your data, take marks off for your analysis, because you just told me you messed it up. Okay, don't tell me you messed it up. Don't mess it up. That's better. Okay, don't mess it up. If you make a mistake, that's okay. Just correct it right then. Right? You want to get that good data. Okay. All right. Questions on lab reports? Okay. So when a lab report is done, I mean, if you're looking at this one right here, this doesn't have anything really in it yet. Okay, and it's already over a page. Okay, once you get this stuff filled in, it could be closer to 2 pages. Okay, that's kind of the expectation. Okay? Is that when you fill this in and you're using complete sentences and you're giving good detail and all of that kind of stuff, that yeah, it is going to be, you know, quite a bit longer than that. Again, you don't have to build the whole thing. It's already partly built. You just need to fill the stuff.
Do you have to like relist your materials in your procedure if you already have them up there?
No. So there is never going to be an experiment where, that like for a lab we actually perform, where you are going to have to do anything with the materials. We do a couple of activities in the bio unit where you have to design the experiment completely. Then you would have to put the materials in yourself because you would decide what they would be. But no, otherwise it's all going to be in there and you don't have to, because this is what you're handing in, right? So all you're doing is filling in the rest of it, okay? And then clicking the turn in button in Google Classroom. Okay, so don't go and like take a whole bunch of time copying this and rebuilding it somewhere else. This is the one I want. Okay, in fact, I won't accept a file that isn't the one I put in there. Okay, each of you will get this that'll say your name, U1 properties of compound. That's the file that gets turned into me, not something else. Okay, if it's something else, I can't go back and check your revision history. Okay, that's an important thing for me to do. Okay? If your lab report suddenly appears in 30 seconds, I know you didn't write it. Yeah, and I do that. Okay, I do go back and check the revision history. Okay, and I've actually had some kids I heard, and they were like, "Okay, we'll just go to the library and we'll copy it all down. We'll just copy yours." So they went to the library, and I went in because it's my file, and I watched them cheat. And then I called them in and went, "Okay, now you guys can sit down in here and do it by hand, on paper, so you can't cheat." Okay, yeah, it's cool. It's cool that you have that kind of ability to check that stuff. Okay, so yeah, make sure that it is this file here. Okay, plus then I can put stuff in this one, you know, as giving you feedback and stuff. So I don't want anything in on paper. Just going to come in this way. Okay, other questions? Okay. I'm going to give you guys like a quick break here, and I'm going to bring some of the equipment we're going to be using over here so you can kind of see what it is. And we're going to talk about some of the tests that we're going to run in the lab for Monday.